Hello, everybody. Welcome back. So this video is coming after I've gone through the whole alimentary canal. So you may have seen the layers and control video, the stomach, small intestine, large intestine. So now I'm going to take some time and talk about some accessory structures, the pancreas and the liver. I'm putting them together because they both secrete their product into the same place, into the duodenum. But they are quite a bit different. So we're going to talk about the pancreas first and then the liver. All right, so here is where the pancreas sits. It's kind of nestled up in the curve of the duodenum. The stomach sits just above it, so it's kind of sandwiched between the stomach and the duodenum and the beginning of the jejunum. So you can see there that it has a large duct that runs through the middle from the tail towards the head, and it drains into this duodenal papilla, which is the papilla means bump, right? So it's an opening that drains into the duodenum to release the secretions of the pancreas. So what are the secretions of the pancreas? Well, there are two parts. So if you remember way back in 232, when we were talking about hormones, we learned the pancreas has two hormones that it secretes, the insulin and glucagon, which are used to regulate your blood sugar levels. You can go back and review chapter 18 if you need to. I'm going to be spending more time on the exocrine function of the pancreas since we already covered insulin and glucagon before. So just to kind of get that out of the way, so that would be our islets right here. So this is the endocrine part. So we're not going to talk about that. We're going to talk about everything else. Like 1% endocrine, 99% exocrine. So we're going to talk about the 99%. So the exocrine secreting cells are called pancreatic acini, right here. These guys right here, they're the little cuboidal cells. And what they do is they secrete a little concoction of stuff that's going to help continue on the chemical breakdown of your molecules. They secrete their products into these ducts, and then all of those kind of smaller ducts will drain into the larger duct, will eventually go into the duodenum. There's a small percentage of the population, like 10% or less. I see my cat on the counter. She's not supposed to be on the counter. So 10% or less of the population may have an accessory duct. But it's just an alternative pathway. They're both going to get to the same point, is the duodenum. So the secretions that the pancreas makes that are non-endocrine are a lot of enzymes and buffer. So it makes a buffer. And buffers, if you remember from the respiratory system, their job is to maintain a constant pH. So if acids show up or bases show up, they can kind of help keep things steady state as far as a pH value goes. So think about why the pancreas might be squirting a big bunch of buffer into the very first part of the duodenum. Well, what's coming into the duodenum? What is the duodenum hooked to? It's hooked to the stomach, right? So the stomach sits right here. That's a really bad stomach. So this has a lot of hydrochloric acid in it. So the chyme that just spent 4 hours churning away in the stomach is mixing up and slowly going into the intestine. The intestine is not made for acidic chyme. It doesn't have the thick mucus like the stomach does. So we need to neutralize that. So the buffer comes from the pancreas and neutralizes the chyme that's coming into the duodenum. All right. It also secretes enzymes. Now we've seen a couple other enzymes. I didn't talk about the oral cavity, but you read about it. So we are getting some enzymes here. I'll put a list up here. So we have amylase. Amylase works to digest starches. It takes large starches and breaks them down into sugars. We have pepsin. Right? So pepsin came from the stomach. It breaks down large polypeptides into shorter polypeptide chains. We also have amylase coming from the pancreas as well. So we have salivary amylase and we have pancreatic amylase. We also have, I'm going to just collectively call them pepsidases. Because I'm not going to make you learn all of them. There's a lot. There's a key one called trypsin. There's a chymotrypsin. proteases. So all of these collective peptidases are there to continue the breakdown of proteins. So pepsin in the stomach, the collection, the family of peptidases. We also have lipase. There's a little bit of lipase that comes from the tongue, but there's pancreatic lipase that breaks down fats. And then there's also nucleases. So these are going to break down your DNA and RNA into your nucleotides. Again, we don't use the nucleotides for nutrition, but we do absorb them and use them as building blocks to make our own DNA and RNA. So here we have a whole collection of enzymes that are being secreted from the pancreas, or the alkaline buffer. So it's going to be probably a pH 8 buffer. And we have this huge collection of enzymes, and their job is to do that, getting as close to that final step of processing as possible. Because remember, the final step was those brush border enzymes that are located on the microvilli of the small intestine. But the amylase, the pepsins, the peptidases, the lipase, this was in the stomach. I wrote it up there because we're talking about enzymes, but it does not come from the pancreas. Pepsin is in the stomach. So amylases, peptidases, lipases, nucleases, these are a whole kind of cocktail of enzymes that are coming out of these pancreatic cells. I'll kind of color them green. So this is going to be our cocktail of enzymes that are then going to be secreted into your duodenum to start mixing and churning with this chyme that's coming from the stomach and do this chemical breakdown. And then the brush border enzymes will do the final breakdown. Okay. So that is our pancreas. Amylase, lipase, nucleases, proteases. Now you might be asking yourself, if all of these enzymes break down organic molecules like carbohydrates and proteins and lipids and nucleotides, how is it that we do not digest our own pancreas? Good question. Well, we have the barrier of the actual duct right here. So I'll delete all these kinds of things. So we have this duct that's going to be protective here, that's going to not allow the enzymes to dig deeper into the pancreas. But we also, a lot of these enzymes are secreted in their inactive form, kind of like how pepsinogen was inactive and then hydrochloric acid converted it to pepsin. A lot of these enzymes are released in what's called a proenzyme form. It's before the activated form. So like pepsinogen is a proenzyme for pepsin. Fibrinogen is a proenzyme for fibrin. Angiotensinogen is the proenzyme for angiotensin. So a lot of times our enzymes are secreted in this inactive form that then once they get into a more appropriate place, then they can become activated. So they're not active when they're being made in the cells. They become activated once they're away from the cells in these protected ducts and into the more protected lining of the duodenum. Okay. All right, so that is our pancreas. Okay. Next, we're going to talk about the liver. The liver is our largest internal organ, so we can kind of see how it sits anatomically in correlation to the stomach and your up under your ribs. We can kind of see it pushing up against the diaphragm a little bit. It does have a few lobes, but I'm going to zoom more into and spend more time on the microscopic level of the liver, because that's where all the functions happen. And the liver has a lot of functions. In the digestive video you'll be watching, it says the food machine, it says about 500. I think your book says about 200 different functions. We're not going to go over all of those. We're just going to kind of talk mainly about the categories of liver functions. Actually, let's do that right now. So the categories of functionality that these cells do, and they're called hepatocytes. So the hepatocytes are the name for liver cells. That's basically what it means, hepatocyte. So one of the jobs that they have is carbohydrate metabolism, and this is listed in your notes. It helps to, it's one of the targets for your insulin and glucagon. So when you have high sugar levels, insulin is released, opens up the doors, allows these hepatocytes to take up the glucose, and they store it as glycogen in a process called glycogenesis. is we are making glycogen, which is a storage molecule of glucose, which is a whole bunch of glucose molecules hooked together. So your hepatocytes are doing that in this time of plenty, if you will, just after, it's called the post-absorptive state, right after you eat. Now, if it's been a while since a meal, and if you remember, we talked about releasing glucagon. So glucagon goes out and primarily targets your liver and tells your liver to release some of that glucose that you just stored after the meal. And this is called glycogenolysis, right? So now we're breaking down. So instead of genesis, instead of making glycogen, we are lysing the glycogen, and that's going to release the individual glucose molecules back into the bloodstream, bringing your blood sugar back up to normal levels. And then the last thing that the liver can do in response to needing glucose is it can actually make more glucose molecules, typically out of fats and proteins. Amino acids and the glycerol in fats can be modified to be turned into glucose. And so this is a long one. It's called gluconeogenesis, right? So gluco meaning glucose, neo meaning new, genesis meaning to make. So the liver can actually make new glucose as needed. So it can store glucose as glycogen, and it can release glucose by breaking down glycogen, as well as generating actual new molecules of glucose. For the lipids, so lipid processing and regulation happens here as well. So cholesterol is formed, the chylomicrons that got picked up by the lymph can be processed, and your fatty acids are also processed here. Amino acid metabolism, so the proteins that are being, the little amino acids that are being picked up in the villus, they go into the liver, and the liver takes up that raw material, and the liver is a big manufacturing plant for a lot of our plasma proteins. So albumins, the complement proteins, some of our other clotting factors, those are all proteins that are made in the liver. So if you're going to be making a lot of proteins, you need a lot of amino acids. So it's going to harvest those amino acids out of the blood and start using them as the building blocks to make your body proteins. The liver also removes waste products, so ammonias and ureas. It can kind of filter and take out of the blood to detoxify. It can store your fat-soluble vitamins. So vitamins A, D, E, and K are fat-soluble, so those can be stored in some adipose, some fat storage parts of these hepatocytes. Your liver also can store some minerals like iron in the form of ferritin. And it can actually deactivate drugs. So a lot of times when pharmaceutical companies are figuring out how a drug works, they have to take into account what the liver does to it, because it can break it down, it can metabolize it, it can change it into an active form. Maybe you take it as an inactive form, and by processing through the liver, it turns it into the active form. So the liver can modify and neutralize or deactivate drugs, and also does alcohol too, deactivates alcohol. So those are some of the metabolic functions that the liver does. It also does some hematological regulation. So these are all just listed in the notes. So we have the Kupffer cells. Let's see if we can find those right here. These little pink guys. Those are antigen presenting cells. So if you have infection in the liver, they're going to find that antigen present on the class 2 MHCs. Synthesis of plasma proteins, I already talked about that with the taking up of the amino acids. This is also a place where your hormones can get recycled out of your bloodstream. So let's say your thyroid stimulating hormone goes out, tells the thyroid what to do. Does it just stay circulating throughout the blood? No, it doesn't. Those TSH levels are going to go down, and the liver is one of those places where that happens. It can also remove antibodies. As your antibody levels wane, it's because the liver is removing those antibodies out of the bloodstream. And it can remove toxins, and unfortunately, if they're lipid soluble, it can actually store some of those toxins in the fats, which is a bad thing. You don't want the toxins in your body in the first place. It can remove some of them, but if they're fat soluble, they'll likely stick around. And then lastly, kind of connecting to our next organ, is the gallbladder. So the gallbladder stores bile that the hepatocytes make. So I'm going to talk a little bit more about this process of bile production and kind of what these hepatocytes do. So I'm going to erase these words here. So we're going to talk about the setup of the liver cells. So we have the liver is broken up into these kind of hexagonal units called lobules. Okay, so that's one liver lobule. And if you'll notice, there's lots of tubes going all over the place. So there's three sets of tubes that are associated with the liver lobule. We have the bile duct, we have the hepatic portal vein, and we have the hepatic artery. They don't have that labeled, but that's going to be this one right here. So those three things are found in what we call the portal area, or sometimes called the portal triad. So here's a triad, here's a triad. The triads typically are found in the corners of these. And so here's a really big triad right over here. So the triads are found in the corners of these hexagonal shaped lobules, and they have one of each of those three tubes. They have a bile duct, they have a hepatic portal vein, and they have an hepatic artery. And then in the middle of the lobule, we have a hepatic vein. Okay, so where does all of this stuff go? So where does the flow go? Where's the flow? Erase all those things. I'm going to go to kind of like a whiteboard screen, and I'll come back to this picture. So let's say I'm going to draw my liver. Okay, I'm going to draw the heart, and I'm going to draw my intestines and my stomach and that kind of stuff, right? So hopefully you get it. We have, in general circulation, we have our aorta, right? And that's going to be bringing blood to our digestive viscera. And then that's going to be draining in capillaries. And if you remember, we had a hepatic portal circulation, right? So that's going to go directly to the liver. And then we're going to drain that back to the heart. So that's our hepatic vein. And I'll label these in a second. We also have, from our aortic branch, we have blood going directly to the liver. So this would be our hepatic artery. This is the hepatic portal vein, and this is the hepatic vein. So the liver is actually getting two sources of blood. It's getting the oxygenated blood from the hepatic artery coming off the systemic circulation, and it is getting the nutrient-rich venous blood coming from the capillary beds in the digestive viscera. So if you got to think back to your blood vessel list, right? So hepatic portal vein was important because it carried all of those nutrients that just got absorbed from the small intestine, which we talked about, the amino acids and glucose anyways, coming into the liver, and we have the oxygenated blood coming to the liver. And then the liver processes all of that and eventually drains it via the hepatic vein back into general circulation. All right, so that's the big picture. All right, so now we're going to take another look at this microscopic view here and kind of see what's actually happening. Oh, let me pick green since there's nothing, well, no, we'll do purple. Okay, so when we talk about the things that are going into the liver, so that's going to be your, let's go down here. So we have our hepatic portal. So the hepatic portal is bringing in nutrient-rich blood, and we have the hepatic artery, and that's bringing in oxygenated blood. So all of these branches off the hepatic portal and the hepatic artery are going to be going through sinusoids. You guys remember sinusoids are the big flattened, kind of really open and loose capillaries. So the nutrient-rich blood is going to be heading towards the central vein. The oxygenated blood is going to be heading towards the central vein. So the blood goes from the portal triads towards the central vein. And in that process, you'll get gas exchange. So everybody's going to get oxygenated. CO2 is going to get picked up. But you're also going to be dropping off and taking out the glucose for the glucose storage. You're going to be taking out the amino acids for the plasma protein manufacturing. So if we look over here, you're going to be seeing kind of this direction of flow from the triads coming to that central vein through these sinusoids. So this is the blood flow from the triads towards the central vein, which is then going to allow, so all of these hepatocytes have a really close connection to all of this blood that's coming in, the nutrient-rich blood and the oxygenated blood. Now for the bile, one of the jobs of the hepatocytes is to make the bile. So they're manufacturing the bile, and the bile gets picked up by these little bile canaliculi here. So the bile then is being made in the hepatocytes and travels towards the portal triads. So we're going to be going this way, right? So all the bile is going to be picked up by the bile ducts. And those bile ducts will be collecting all of that bile, draining it, and eventually storing it into the gallbladder, which we'll be able to see on the next slide. So I guess the takeaway for this is to just be sure you can understand the circulation to the liver, right? So hepatic portal vein is carrying nutrient-rich blood to the liver. The hepatic artery is carrying oxygenated blood to the liver, and that is going to be processed by the hepatocytes. Well, at the same time, it's doing all those other jobs we talked about, but it's making bile, which are being picked up by the canaliculi, being drained into the bile ducts, which will eventually go down into the gallbladder, which we'll take a look at next. So here's a close-up view of our gallbladder and the duct system, and it comes into close connection with the pancreas, which is why I'm kind of talking about everything pancreas and liver together. So here we have in our flow of bile coming from the liver, it is drained by these large hepatic ducts. We have a left and a right hepatic duct. So all those bile canaliculi and the bile ducts and all of those lobules we just looked at are all kind of fusing together and forming into these two major ducts. The left and right hepatic duct drain into the common hepatic duct, which then drains into the common bile duct, which eventually makes its way down here to that duodenal papilla, right? Down here. So now bile is going to be mixing in with some of those pancreatic juices. But that opening into the intestine is not always open. If we take a look, this is the opening down here in this lower picture. If you'll notice, there's smooth muscle around it. It's a sphincter. It's called the hepatopancreatic sphincter. And during non-digestive times, when you're not active in parasympathetic activation, that's closed. And so you're not secreting your pancreatic juices into the duodenum. You're not secreting bile into the duodenum. It's a closed door. It's going to get some signals to open up. We'll see that with the hormones a little bit later, but it's closed. And so if your liver is continually making bile, but the exit door is closed, what's going to happen? So it's going to be made here. It's going to go down here. The door is closed. It's going to get backed up. The liver can't go back up into the liver. That's too high of a pressure situation. But we have a nice big empty gallbladder. So it goes through the cystic duct, goes to the cystic duct and starts filling up bile in the gallbladder. So the gallbladder is a storage tank for the bile that the liver makes. And the liver makes pretty dilute bile. It's not super concentrated. So when the hepatopancreatic sphincter is closed, the bile kind of backs up to the cystic duct, hangs out in the gallbladder. While it's sitting there, it becomes concentrated. So some excess water gets taken out and all that's left is the concentrated bile salts. So then when the hepatopancreatic sphincter opens up, you're gonna have concentrated bile secreted into the duodenum and not the dilute stuff. So what does bile do? We didn't really talk about that. Bile is an emulsifier. It takes big fat droplets and breaks them down into smaller fat droplets. It's not an enzyme. It's not breaking bonds or making bonds or chemically changing the fats. It's just taking a big blob of fat, and breaking it down into smaller blobs of fat. Same amount of fat, but what do you get? You get more surface area. And so now the lipase, that pancreatic lipase that's coming from the pancreas, will have more surface to attack and actually do break up the fats into your triglyceride might be broken down into your individual fatty acids. So that's the job of the bile, is to emulsify fats, to just physically break them into smaller pieces. Okay. So this is where the secretion of bile and the pancreatic secretions kind of come together, is they fuse here in this this duodenal ampulla, and the hepatopancreatic sphincter regulates when that opens and closes. And that is actually regulated by hormones, which we'll see when we go over the hormone lecture. All right, so that is our pancreas with its secretion of buffer and enzymes, and all the many functions of the liver and the production of bile, and then the function of the gallbladder storage for the bile and how it works together with the pancreas to secrete stuff into the duodenum. All right, I think I got one more mini lesson for the digestive system when we're going to talk about hormone regulation and absorption, kind of wrap things up. All right, I'll see you next time.